Greetings, everyone. Welcome to the presentation today about the All of Us research program, a massive research effort underway here in the United States. It's my pleasure today to talk to you a little bit about the program, give you some background as to what some of our programmatic values and goals are, looking at what this program is doing differently than some of our traditional biobanking studies. Then we're going to spend a little bit of time talking about the value that we hope to bring to the research community through the All of Us research program. After that, I want to talk a little bit about our cohort structure and how that's been going, give you some updates, and just talk about really some of the pros and cons for both of the cohorts that we have. We're also going to take a look at some of our data collection methods, and specifically taking a close look at biospecimen data collection, because that here at Mayo Clinic as the biobank for the program, that's really what our main goal and focus is as part of the program. We'll also spend a little bit of time talking about, of course, because this program is so large, it's oftentimes difficult to talk about it and represent it well in a short talk such as this one. So really, another focus of today is really to speak about the COVID-19 impact that it had, or the impact COVID-19 had on our program, and some of the response actions that we have, and kind of where we are today. And then lastly, kind of some conclusions and why I was asked to talk today through our relationship with DNA Genetech, talking a little bit about what they have brought into our program as a vendor, and some of the challenges that they've helped us overcome. So without further ado, let's get into the presentation itself. So what is the All of Us research program? This is a historic longitudinal effort to gather data from 1 million or more people living in the United States. And the goal is to help accelerate and improve health. They're trying to take into account individual differences such as lifestyle, socioeconomics, environment, and biology, and hopefully help researchers uncover paths towards delivering precision medicine or individualized prevention, treatment, and care for all of us, hence the name. So this program itself is really one of the foundational components to the larger precision medicine initiative. The Precision Medicine Initiative was launched in 2015 by then President Barack Obama, and was really introduced to the United States and to the world during the 2015 State of the Union address. This has really been spearheaded and coming from our NIH director, Dr. Francis Collins, who has the quote here, "The All of Us is really one of the most ambitious research efforts that's ever been undertaken in our nation here." So very much so a pleasure to be involved and to work so closely with a program that I think is really near and dear to everyone's heart. So if we take a look at really the next set of slides here, where we talk about really the objectives and the missions, and also looking at some of the core values of the program, again, really resonates as a citizen and someone who benefits from a great healthcare system, and trying to address some of the inadequacies that we have today. And I truly believe that this program is one of the key foundational components to get us there. So as we look at it in the middle, the mission really is to accelerate health research and medical breakthroughs, which will enable individualized medicine. So how do we do that? First and foremost, it's really creating relationships that are meaningful and can be sustained by seeking out 1 million or more participants as partners in the United States from all walks of life, and all different types of backgrounds, both geographically, socioeconomically, racially, ethnically, whatever. We really want this program to represent the diversity that exists in the United States. After establishing those relationships and building trust with our participants, we hope that this can help create really one of the largest, richest biomedical data resources that's ever been known, with a key focus on making it accessible, safe, and as free to use as possible. Once this data set has been created and is available to the public, we really hope that this helps catalyze a robust ecosystem of researchers and curious individuals who can look at this information and help use it to create new theories, new hypotheses, which can eventually lead to discovery. And as you can see, represented by the diagram here, this will hopefully be a virtuous cycle, continuously evolving and growing as the research community continues to evolve as well. Next here is just a series of bullet points that highlight some of the values, the core values of the research program itself, and some of the things that we are telling our participants, and what drives a lot of the decisions, what drives a lot of our policies and procedures are really fundamentally rooted in these values. So just to walk through them real quickly, open participation is a very fundamental component. And we want the participants that do join our program to represent the rich diversity of the United States, as I previously mentioned. One of the key things that I have always appreciated is the use of the term participants and partners instead of subjects or patient. We really truly want everyone who joins this program to have an active say and to be involved in the research that is being conducted using the samples and the data that the program collects. So viewing them as partners and participants has a much different undertone than the word subject. So that's been a key sort of theme as the program has started recruiting and collecting samples from our participants. Trust, as I mentioned during the last slide, we're trying to really build out a good set of participants that we can engage and we can hope will keep coming back to us for different projects, for serial collections, for a different survey that we would like them to complete. We want to make sure that every participant we have will come back. And in order to do that, trust is imperative. So how do we get there? And one of the key things that we've learned is transparency. So making sure that participants are aware of programmatic policies and decisions, keeping them informed on personnel changes at highest levels of leadership within the program, and making sure that all of the data security infrastructure that is implemented is of the highest value and the latest and greatest in the industry. Giving participants access to their information. This has been another really key selling point and one of the very attractive components of the program for participants. And that is that, in a lot of traditional research projects, the subject or participant comes in, they donate a set of data or material, and they don't really hear back from the program. They don't know how that data was used. They maybe they're running a simple blood test on a certain specimen. It seems like a reasonable ask for those results about someone's health, despite it being conducted in a research setting, to still be accessible and available to that participant. So that's one of the things that this program hopes to enable, is the ability for any specimen results that are derived, or results that are derived from specimens in particular, that we have a way to funnel those pieces of information back to the participants. Also here, the accessibility of the data that is collected, making sure that it's broadly available. And also mentioned just a second ago, security and privacy will be absolutely imperative in order to maintain those trusting relationships with our participants, and also to make sure that the data that is collected is held responsibly and not compromised in any way. So this has actually been a huge hurdle for a lot of participants who are wondering about joining. They really want to make sure that their information is secure. And it's oftentimes difficult to really explain to a person who doesn't have a data science background or an IT background all of the incredible measures that are being implemented by the program. And so despite our best efforts, oftentimes this does happen to be a reason why someone isn't quite comfortable participating yet. But we hope with continued education and continued progress and work that we will continue to overcome this hurdle and get all the way to our goal of 1 million participants. All right, so what makes all of us different than a traditional research study? So of course, the first and foremost, you have to mention the scale of the program. So 1 million people, that's a gigantic cohort, a gigantic set of individuals that we can study. And getting that many people, without really even trying, will naturally lead to diversity of demographics, geographics, medical backgrounds, and history. But I think you'll see, or you will notice if you do any research on the program, there has definitely been a concerted effort to ensure that the underrepresented individuals and participants in biomedical research are represented during this effort here for the All of Us program. The data types, which we'll look at in the next couple of slides here, are very diverse in what we collect and what we look at. And the ability to collect it longitudinally will hopefully allow for additional translative research activities. We touched on bullet number 3 quite a bit on the last slide, but really focusing on participants as partners. And this goes a long way. We have some direct examples of this, having our partners as a part of governance. There is actually a participant advisory board that we call it. So these are people who heard about the program, joined, felt very passionately about it, and actually had the credentials and the time to serve the program almost in a full-time capacity as an advisor, really representing the rest of their community and the rest of their fellow citizens as the program looks to make progress and develop new policies, again, as research projects and proposals are pitched, and as we continue to move forward into new aspects of the program. And again, making this a national open resource for all. So looking at the data, making it available publicly, and making the tools that are utilized to view and analyze the data user-friendly so that it's open to as many people as possible. The next series of slides here, I want to talk a little bit about the value that this, or expand upon the value that this will hope to bring to researchers. Not only the what, but the how. So let's take a look at this. So participants start by being recruited or learning about the program and deciding to volunteer. They share most of their data online through lots of surveys. They also include the option to share their electronic health record, which is digitally transferred to the program's central database. And when eligible and they've met all of the preliminary requirements, participants are also asked to provide physical measurements and biospecimens, which are captured traditionally into a clinic, which we'll talk a little bit about in a few slides. The data, as I mentioned, is transferred to our secure central database and is protected to the highest security standards that the industry has to offer at the moment. Physically, the samples that are collected, the biosamples that are collected, are then shipped overnight to the Mayo Clinic in Rochester, our biobank for the program, where we process and store the samples in our, most of the samples in our robotic freezers in a very secure manner. And completely de-identified, I should mention. The only ID that's on a tube is a barcode, and you have to travel many data sharing linkages away before you're ever even closely able to decide or determine who that sample belongs to. So really the anonymous factor of each of the samples in storage is also a way for us to build trust and encourage participants to donate their biosamples to us. So after data is collected and shared and stored into this data repository, they do a bit of data cleansing before they move the data into what they call a curated data repository, which is used to create this researcher hub or this researcher tool where we actually bring the researchers to the data as opposed to bringing the data to the researchers. So this is, again, another way that we hope to catalyze new ideas and help researchers form collaborations together and work on things that they may be interested in, all in one location in this central data hub. As we utilize specimens, we synthesize survey results, synthesize EHR data. Participants will constantly be fed information and the latest news about what's happening in the program, again, all coming out of this central data repository that is also connected to our participant-facing technology. So again, keeping our participants involved and informed along the way is a key aspect of this program. And then, of course, at the end, after researchers have dug into the data and looked at it, they are able to share their discoveries. And hopefully, again, this continues to catalyze more and more attention to the platform, bringing more and more people, and just accelerating the amount of breakthroughs and the amount of discoveries that we can make. So again, what is the promise to researchers? So I think here we can look at a lot of things that are very helpful. The first of which is the time and the money it takes really to set up the infrastructure needed to collect from a cohort, and to recruit subjects who may be eligible for your project. Here we're hopeful that we're going to have a wealth of information that's sort of ready to go and ready to utilize, almost like a commercial bank where you deposit money and then you can take it out and use it at a later time. That's really the idea behind this biobanking aspect, is putting stuff away that we are pretty confident we're going to use, so that when a new prevailing disease or a new research method is available, instead of having to put all the legwork into collecting the samples, we're hopeful that oftentimes we'll have them all ready to go, including the participant data, the surveys, the EHRs, and all of that other corresponding information, including the biospecimens, that we can ship out to any given researcher, and start making discoveries more quickly. Engaged participants, I've mentioned quite a few times today, building that trust, making sure that they're coming back and they're staying in touch with us, will also help drive participation in ancillary studies or even potentially clinical trials. We talked a bit about sharing, workspaces and data analysis within that researcher hub. Again, a lot of this, the researchers may have the option to make it public, so it can be viewed and looked at by others. Again, forging some new relationships that may not have existed before. Let's go to the next slide here. So what are some of the questions that we might be able to answer? So you can kind of get a look here, but here's a good example. Can we develop better pain medicines that aren't addictive? Can we drive sustainable interventions that support health equity? Can we develop cancer cures that will work the first time, so that we can skip painful trial and error chemotherapy? I think there's a lot of key research questions here that are oftentimes difficult to address, and oftentimes due to the size of, or due to the lack of size of your cohort. Again, so these are some of the things that we hope to accomplish, having this gigantic data set and this gigantically engaged cohort at our disposal. So some other bullet points here on scientific opportunities. NIH leadership feels strongly that this will help develop quantitative estimates of risk for a range of diseases by integrating environmental exposure and genetic factors. So that environmental exposure piece really comes from survey data. The program also has intentions and has started piloting some wearable data trackers that participants can consent to and participate in, so sharing their data, for example, from their Fitbit, or eventually I think the program also hopes to engage the Apple Health Kit and allow participants to consent and share that data with the program as well. Again, bringing in just another layer of data that many programs are unable to collect. So the next bullet here would be identifying causes of individual variation in response to commonly used therapeutics, or what we call pharmacogenomics a lot. New biological marker discoveries, developing solutions for health disparities. Again, I think that's really one of the key aspects of this program, and something that I'm very proud of to be kind of involved with and observing, is the concerted effort made by the program to ensure that participants who have traditionally been underrepresented are included in this project. So let's talk just briefly about the cohort structure here. So the main difference here, so we have two distinct cohorts within this program. The first of which we kind of call our HPOs, or healthcare provider organizations. So these are participants who will most likely be directly recruited from their existing medical healthcare provider. This would include regional medical centers and academic healthcare centers. federally qualified health centers, and we also partner with the Veteran Affairs clinics. Secondly, we've got another cohort that we consider direct volunteers. So these would be individuals who may not be a member of the partnering institutions at an HPO, but through national advertising campaigns, word of mouth, whatever the case may be, they hear about the program and really want to participate. So this happens to be a very digital approach to joining the program, and has proven to be successful, and also really helps us reach our goal of collecting and involving participants from anywhere geographically in the United States. So here is a great summary of the all of the data aspects that are collected in the program. I have touched on almost all of these thus far, but really the basis of what we're looking at is some preliminary data that is mostly digitally collected and captured either through the AHR consent and transfer and through a series of surveys that are developed and delivered through the participant app. Next, here highlighted in green, is typically a in-clinic or in-person visit where we do some data collection, looking at physical measurements and biospecimens. And then, as mentioned just a slide or two ago, the ability to consent to bring wearable technology into the program, a more passive data collection approach. This slide here really just breaks down our current blood and urine protocol and what the parent collection tubes that we are collecting and what that's translating to at the biobank when we process and store all of the specimens. I believe right now we're sitting at approximately 34 to 35 aliquots that end up in storage per participant when we get the full blood and urine donation. As you can see, a wide range of specimen types are being stored and banked for the program, including DNA. This is a slide that briefly describes our approach to saliva collection. Right now, the program still heavily favors blood collection as our means to obtain DNA from participants. But as we are well aware, it's oftentimes difficult to get blood on everyone, or in particular this case, a lot of these are healthy individuals in the United States and may not want to be poked with a needle. And so therefore we've got a backup approach that allows us to still obtain DNA. And that is really where our relationship with DNA Genotech begins. And this has been something that we have done from the very beginning stages of the program, again, in an effort to ensure that we have DNA stored for nearly every single participant that joins. So this is just a quick overview of some of the advantages and what we think really has been working well from the healthcare provider organization cohort. Before COVID-19, we had a peak where we were really seeing almost 500 to sometimes 600 participants being collected each day. And that was being accomplished primarily through our healthcare provider organizations. And we had roughly 200 active accounts or collection sites across the United States. These are definitely more brick and mortar facilities with robust IT, laboratory, and clinical infrastructure. And really, in order to get them stood up and prepared to see participants, required a pretty rigorous onboarding process that included on-site training, virtual training, some testing exercises, and logistics arrangements. Some of the strengths within this cohort, I think, are really the ability to build trust with their participants, because many of these individuals are already members of that healthcare institution. So they've established trust through other means. They're definitely able to have high throughput at any given site, which often leads to logistics efficiencies and cost savings for a lot of our transportation fees. And it's also pretty proved to be a simpler approach when it comes to change management and protocol revisions. Much easier to educate and retrain and pivot with this group of individuals given their physical infrastructure and their staffing models. The second cohort we mentioned was our direct volunteers, which are supported by a national central coordinating center who then subcontracts out with several vendors and partners. And again, their goal is to reach participants who are interested but do not have direct access to one of our healthcare provider organizations. So this has really been accomplished thus far through event-based marketing, targeted digital marketing in certain regions of the country, and then providing some in-home collection options, blood and saliva as well. We work with a variety of different phlebotomy providers, including those that do in-home collections. And training digitally is definitely the preferred route here. A lot of these contractors and sub-vendors work on many, many projects and rely heavily upon one-page documents to kind of go in and out of their visits. So education has definitely been a bit of a challenge here. However, some of these strengths are, it really opens there and it allows the program to reach that goal of inclusivity. I think this group is very agile and adaptive because we can basically change a one-sheet instructions for the phlebotomists and off they go. And there's a less rigorous onboarding process due to the lack of physical infrastructure. The COVID-19 pandemic, of course, impacted our program very heavily because this is kind of like a lower tier research program. We scaled back and essentially shut the program down for approximately 6 months while we reassessed and sort of distributed some of the resources within the program towards COVID-19 research efforts. But it didn't mean we came to a complete halt. So in-person activities were completely shut down, but remote work was definitely implemented, including some retention phone calls and looking at some different surveys that our participants had available to them, and helped the program kind of catch up on planning and self-assessment activities. What has been the response? Getting back to normal. So I think one of the very first things we were able to reinstitute in terms of specimen collection was an in-home saliva collection process where our vendor, DNA Genotech, mails out a custom kit to our participants who are eligible and interested. They provide their saliva sample and then mail it back via USPS to our biobank where we extract DNA and continue to move forward. So this was a huge component and really allowed us to sort of keep the momentum and keep the attention on our program without completely stopping. Since late 2020, like the fall, we really started to kind of re-onboard and reinstitute in-person activities again. But I think one of the lessons learned, in my opinion, is a program of this size and scale, it's kind of difficult and painstaking to wake it back up, right? So we had to do lots of planning for new safety protocols. Staffing had to be examined and adjusted. Massive retraining efforts, and then restarting our logistics again was also a pretty large undertaking. So how has this relationship with DNA Genotech helped foster and really help our program? So I've mentioned once or twice, the participation being open to everyone is a mission critical task. And without our ability to utilize USPS and collect saliva, there are many, many participants that are just very, very difficult to reach through traditional blood collection pathways. Having DNA from the entire cohort, as I mentioned earlier, when blood is not an option, saliva has proved to be a really reliable backup. There's great flexibility in the laboratory because of the stabilization liquid that allows us to sort of batch our samples and not have to extract them the day we collect them. Logistics costs when reaching individual participants is certainly a bit lower using USPS. And average DNA yields tend to be on par with our blood collection. There are, of course, a few challenges. So here's just a brief overview of some of the data that we have. So recently we switched to the Perkin-Elmer ChemMagic 360 extraction module. And since then we have run approximately 80,000 samples through it. And for our whole blood EDTA, we are looking at a failure rate of about 0.4%, and for saliva about 6%. And you can kind of see the criteria for failure listed there. Of course, there's some additional challenges, including the unsupervised in-home collection. So we're really dependent on our participants' ability to read instructions and follow the protocol. Participants oftentimes do not like their sample to be de-identified. So sometimes, even though it's against the protocol, we have seen where participants will share their name and information in the kit when we receive it, and then we have to go through an entire reporting process. Person is not being trained on using the device or practice using the device, so they've kind of got one shot to get it right. And if any mistake happens, there is potential contamination or issues with the sample. Because of the different durations of time in transit for the sample. It's sometimes difficult to plan for how many samples might show up in the lab on any given day, which kind of corresponds to this bullet point here of an effective electronic ordering system. So we've had difficulty deciding, okay, when do we want that sample order submitted so the lab knows about it, knowing that, some participants may not ship their sample at all, some participants may not ship their sample for several months, and then again, incorporating that variability in the USPS transit time has also been a bit challenging there. So with that is the conclusion of my presentation. I know I went a bit over time here. I'm not sure if we'll have time for question and answer, but happy to do so if we have any questions in the chat box. So I'm seeing a question about the partnership with Apple Health. Unfortunately, I'm not so sure about that. Again, our scope of this program, from my perspective, working at Mayo Clinic at the Biobank, is really hyper-focused on that biospecimen collection, processing, and storage, and utilization of the specimen. So not quite as in tune with the other data collection aspects of the program. That really is managed by several other partners within the project. Yes, of course. Well, thank you again, everyone, for your time today. I wish we would have had a bit more time. There were a couple other good questions in there, but I really enjoyed talking to you today about the All of Us Research Program, and I hope you found this helpful. Thank you.